This is the laboratory activity for the gram stain, and by this point, presumably in lecture, you learned about the gram stain, and it was on the first exam. The problem with that is that if you only learn about it in lecture, you only learn how it is supposed to work and what it is supposed to tell you. And if you actually perform it in a laboratory, you have to also perform all the skills that you need to successfully do the technique, and there are a lot of those. So it's tricky the first time you do it because there are so many different things involved, and none of those come up when you read a description of the test and how it's supposed to work. So let's go through this. The first thing is just to review what the gram stain is. It's supposed to tell us whether bacteria are gram positive or gram negative. And this table kind of walks you through the way a textbook shows it and how it is supposed to work. So here in this column, you're looking at cell walls, and here on this column, you're looking at what a cell would look like under the microscope. So on this side, you have a gram positive cell as it goes through all the different steps with all the different dyes and liquids being poured on it. And over here, you have a gram negative cell as it goes through all the different steps and you have the different liquids poured on it. Over here, you have a cell membrane, a cross-section of a cell membrane, and then a peptidoglycan layer, a thick layer for gram positive, and a cell membrane, thin peptidoglycan, and then outer membrane for gram negative. Yellow indicates that it really is the cell membrane. It's what makes a cell waterproof. The outer membrane of a gram negative isn't really waterproof. It lets molecules in and out. It just kind of restricts their movement and makes a semi-protected space for proteins in this periplasm here. So the first thing that we do in a gram stain is we put crystal violet on the cells. And at this point, we don't know whether it's gram positive or gram negative. And the cells are microscopic, so they look like a little haze on a microscope slide once you add this dye. Then you add grams iodine, the mordant. And what this does is form complexes of the crystal violet. So the idea is that you already have molecules of crystal violet in the peptidoglycan layer. And this mordant networks them and makes them stick better. And if you read about like how to dye fabrics, how to make fabrics whatever color you want, mordants are an important step, and they keep the dyes from being washed out of the fabric. And it's the same kind of idea here. It helps the dyes stick. The most important step here is then this decolorization step, where the decolorization solution is poured on. And what this is going to do, since this is like ethanol or a mixture of ethanol and acetone, and it's going to dissolve the outer membrane of gram negative cell, and it's going to strip the crystal violet out of that peptidoglycan layer. And what that's going to leave behind is the gram positive cells are going to still have the color they got from the crystal violet and the iodine. The gram negative cells have now lost all their color. So if you were to look at these under the microscope at this point, which you would not, but if you were going to do that, gram positive cells would look like this and gram negative cells would be colorless and invisible. You might be able to find them. I don't know. Sorry about jiggling the screen. Whatever. The last step is the counter stain, Safranin, and this is a pale pink stain or a pinkish orange stain. And you would pour this on just to make these cells visible so that under the microscope you could see for sure that you had cells there and they really are pink, gram negative. This dye is too pale to interfere with the appearance of a gram positive cell, which would be dark purple or purplish blue. So what you'd be seeing at the end of this stain, once you've gone through all these steps, you'd put it under the microscope, and if your cells are gram positive, they've got all this crystal violet in their peptidoglycan layers, so they would look purple, and if your cells are gram negative, they would have lost it, but you would have counterstained with safranin, so that safranin would be there, and they would be pink. This is how it's supposed to work, and then then you know whether your cells are gram-positive or gram-negative, and that narrows it down if you're trying to identify them. And it already tells you quite a bit about them. So in order to do this technique, you need to have a microscope slide with bacteria on it, and you need to apply these liquids for defined amounts of time in defined ways. And there's more to it. The first thing you have to be able to do is find bacteria under a microscope. And if you've never done that before, I assure you it is harder than histology. It's much, much harder. It takes people about 30 minutes the first time they're learning how to find bacteria under the microscope, just because they don't have as much color as you get from a stained tissue section. And so they're pale and they're much too small. So if they're out of focus at all, they completely disappear. So it's tricky to do this. And this would be a big part of learning the gram stain. You'd learn that at the same time you're learning to use the microscope. And so if you've done histology, you're off to a good start because you know how to use a microscope, but you'll have to unlearn some bad habits and learn some good ones to be able to see this. And a thing that you wouldn't know how to do if you hadn't done it before would be putting microorganisms onto a microscope slide and attaching them so they don't wash off when you pour the liquids over their surfaces. That's a tricky thing. You have to affix them to the slide, and that itself is a tricky step. And probably the biggest challenge is growing the cultures without contamination. It's using aseptic technique to grow up the bacteria you want and put them on the slide and be confident that the ones you're looking at are the ones you're interested in. So if you work in a clinical lab and a patient's sample is sent down to you and you're supposed to look at the bacteria, well, you want to make sure that you're looking at bacteria from that sample, not from dust in the air, not from your skin. And those are common sources of contamination. And it's entirely possible that if the patient has a slow-growing pathogen, any contaminant will grow faster, and you'll end up doing experiments on your contaminant and not know that it's a contaminant. So that aseptic technique, using the inoculating loops and Bunsen burners and being very careful to avoid dust, that is a critical skill you would learn if you were in a microbiology lab. So I'm going to take you through 3 different problems that come up with a gram stain, and that'll be that. The first problem is that the decolorizing solution, so the third step, it decolorizes all cells. So it's removing the crystal violet from gram negatives, but also from gram positives. The difference is that it's a slower process to remove the dye from the gram positives. So you'd only expose your mystery cell for a short amount of time, a very defined, very repeatable amount of time. And that should be the amount of time it takes to lose all the dye from a gram negative, but retain it for a gram positive. That's the idea, is you decolorize long enough to do this without losing all the dye from the gram positive. And again, that's like ethanol or ethanol and acetone. So it's a solvent that is it's dissolving lipids, but it can't do anything to peptidoglycan. So if you were to decolorize for too long, and this is definitely a thing that new gram stainers will do, if you leave the decolorizer on too long, it will remove all the dye from all the cells, gram positive or gram negative. That makes this a tricky technique because the timing is critical and you have to do a lot of practice to control the timing. You might have an excellent stopwatch, but that doesn't mean you'll be able to physically control which liquids are touching the cells. That's a skill you'd acquire through practice, and it's really a thing that we would all focus on in the lab because that's the thing we can control. So what I wanted to show you with this diagram if we compare it to what you'd see in a textbook, so if we go back here, what they show you is once you've added the iodine, the crystal violet sort of swells and becomes darker. And that does happen. That happens in both types of cells. And then once you decolorize, you lose some of it. But they only show that happening on this side, and that's silly because it happens to whatever cell is touching the ethanol, whether it's gram-positive or gram-negative. And that's what I tried to show with this altered diagram, where during the decolorizing step, we are losing dye from both types of cells. And then at the end, when we use safranin, well, this shows that it has lost some of the crystal violet. There is a lighter area on the surface, and that's what I was trying to show you through that comparison. Another problem that can be a bigger problem is that when you grow up cells, and if they are old, if they're in later stationary phase, so if they grew up overnight and you gram stain them late in the afternoon, some of the cells will behave differently, and gram-positive cells might appear gram-negative. Those things would happen. So it's a tricky thing to time this this technique. And if you don't know how fast a cell doubles, you can't plan ahead. The last problem is one I've mentioned before. A contaminant that falls into a culture could potentially grow faster than the cells you actually want to study and potentially take it over. One example of this is yeasts. If there's, yeasts. If there's Candida, for example, or even some molds, they grow very quickly and they are tightly associated with people and skin. And I have seen students think they are doing a gram stain on bacteria and they do a gram stain on yeast and it doesn't work. And so they spend all this time trying to answer a question about a bacterial unknown species for their class. And the whole time they are working with yeast or mold or something completely unrelated, completely irrelevant. And that just happens if they make a small error with aseptic technique when they are starting the culture that they're later going to analyze. They don't have an easy way of realizing they made that mistake. And so aseptic technique is a set of techniques people would have to use very well. And you have to have a lot of experience to know when you might be in danger of having that contamination. And so these are the three biggest problems that students might encounter when doing the gram stain in a laboratory. And it's a bummer that you don't get to do it because in order to do the gram stain, you really do put together a lot of different skills. And once you've done it successfully a few times, you really have skills in the microbiology lab that you can use for other purposes. So if you ever get a chance to go into a lab and learn how to do it, I'd recommend it. All right, have a good day.